Yeah, let's get started. It's top of the hour and we are recording. So I wanted to welcome everybody to this webinar organized by the Society for Immune Polymorphism. My name is Martin Myers, and I have a couple of quick announcements. The first is the topic of this webinar is the human CD1 lipidome. And this webinar is organized kind of in advance of this hackathon, which is taking place in September 17th through 19th. That's been organized by Willie Hildebrand and his group. And we have, as with most hackathons, a set of topics that we want to focus on. And we have this peptide ligands for HLA and a topic of non-peptide ligands. And, MR1 and CD1 are two of the systems we want to look at. And so as sort of background to this, because I'm not as familiar with this side of the ligand story, we've invited two speakers to come talk about it. And the last housekeeping item is I just wanted to let everyone know Society for Immune Polymorphism exists. We are incorporated this year. We have a website, which is immunepolymorphismsociety.org. And if you'd like to join, there's the tiny URL, join the SIP, and you can stay in contact with us and so forth. So now I'd like to hand it over to Willie Hildebrand to host the rest of this webinar and to introduce the speakers.
Thanks, Martin. It's a real pleasure to be working with the Society for Immune Polymorphism today. I know many of the members of the Society, and it's a great pleasure to interact with them. So we're going to have, as Martin said, we're going to have two speakers today. The first is Dr. Christopher Thorpe, and he indicated that calling him Chris is fine. Let me pull up his bio real quick. That's not the right bio. Let me go to this one. So Chris is an Embl Maurice Sklodowska-Curie, a Rees Fellow, working at the Embl European Bioinformatics Institute in Higston with collaborators at the University of Oxford. Rees is an EU-funded program aiming to develop new technology and methods in life sciences and develop skills related to running successful research infrastructures. His role focuses on building research tools and curated data sets for fellow immunologists and co-designing and testing these on research applications with his collaborators. One example is the human CD1 lipidomic database built in collaboration with the laboratories of Hashem Cooley and Graham Ogg in Oxford and Branch Moody at Harvard. Originally a chemist, Chris holds a PhD in structural and computational immunology from Birkbeck College, University of London, and is a Fulbright scholar. He returned to science after a successful stint in the technology sector, where he helped set up Biomed Central, the Guardian Open platform, and the government digital service. So without further ado, Chris, it'd be great to hear your presentation titled The Human CD1 Lipid Portal.
Lovely. Thanks so much, Willie, and lovely to meet you all. Let me just share my screen. We can get going. So hopefully folks can see the slides. Apologies if my voice goes a bit. I had COVID last week and I'm on a hotel Wi-Fi, so it's not exactly the best way to do a talk, but we'll muddle through and see how we go. So I want to talk a bit about a preview that we're building of this portal. We'll have it ready for the hackathon in a sort of complete version that folks can play with. But I wanted to really introduce a tiny bit what CD1 was and a bit about the lipids that it binds. And I'll leave the main meat of that to Xiaoshang, who's been doing amazing work on this and knows it far better than I do. So I'm an MHC peptide person converting to sort of CD1 and lipids. So I'm the sort of test case for lots of this information, and much of the information in the presentation is going to be pulled together as a tutorial, which will be on the website to try and ease the on-ramp for people who are interested in this. And we've got sort of three main sort of target audiences for the portal. One is for sort of people who are, existing immunopeptidomics people who are interested in lipid presentation and trying to understand what you can do with the data from a machine learning perspective or whatever. We're also interested in, MHC folk who are sort of curious about these lipid presenting molecules and who want to learn more about them. And then finally, we've got another audience, which are people who are well versed in lipids and lipidomics in general, but who are new to the immunolipidome as presented by CD1. So what are CD1 molecules? Well, they're non-polymorphic MHC class 1 homologs, and they bind lipids rather than peptides. They bind the lipids in what we normally call the peptide binding groove. And there are 4 isoforms. So rather than polymorphic forms, there are 4 isoforms: CD1A, CD1B, CD1C, and CD1D. You'll also occasionally hear about CD1E, which is a soluble isoform, which there isn't at the moment, as far as I'm aware, lipidomics data for. They all differ slightly in their binding cleft architecture, just as you have polymorphisms that tune the binding site. So these different isoforms have different binding properties. They also have different tissue distribution, and there are different cells which they interact with in terms of sort of T cells. So some will interact with alpha beta T cells, some will interact with gamma delta T cells and NK T cells. If you look at them structurally, and as a structural biologist, this is the way I always sort of try to think about these things. They look a lot like class 1. They've got a similar rake of the alpha 2 helix that class 2 has. We all know that class 1 and class 2 are pretty similar in MHC. And if you were to sort of look at them without knowing that there was a lipid in there rather than a peptide, you'd probably think they were the same thing. If you look at them from above, though, they're really quite different. So instead of having a sort of like a bowed open cleft where the peptide sits, as in class 1, which you can see over here on the right at the top, and for class 2 just below it, they have a much more sort of closed groove. There's a salt bridge which holds the 2 alpha helices together and holds the lipid tail in place. If you look at the surface plots for them, again, we're all used to seeing sort of class 1 and class 2. So class 1, again, is where my mouse pointer is. It's got the walls at either end of the peptide binding cleft that prevent the peptides from extending at either end, but bulging in the center, whereas class 2 has the open peptide binding cleft. But all the different CD1 isoforms have a very different peptide binding cleft. There's a shelf, which I'll talk a bit more about in a second, which closes off what you would normally think of as the N-terminal end of the binding cleft, which is the end which holds some of the lipid tails. And there's a sort of small opening, slightly larger in CD1B and CD1D than in CD1A and CD1C, where the head group of the lipid pokes through. And just to give you an example here, these are the usual peptides that we're used to with class 1 and class 2. And these are the CD1 isoforms with a lipid bound there. And the T cells can actually see the head groups of the lipid that protrudes through this hole. If we look at it from the side, this is sort of a side slice through the binding groove. You can see this sort of shelf that sits over the top of where the sort of like we're used to the N-terminus of the peptide binding. And that creates a really enclosed space for one of the sets of tails for the lipids. And if we superimpose the lipids, you can sort of see that actually where there is this opening, the head groups, the lipids can protrude through and are accessible for the T cell receptor to see. If we take a look at the lipids themselves, they're really quite a variety of them. It's a very hydrophobic binding site for CD1 for the different isoforms. And whereas with class 1 and class 2 molecules, we're used to seeing lots of water molecules in the binding groove to sort of provide a packing and spacer function. With CD1, we see occasionally with certain isoforms, with 2 in particular, CD1B and CD1C, we see these sort of scaffold and spacer lipids. So in yellow are the antigenic lipids. So these are the ones that vary. And then for some isoforms, they have a set of different spacer or scaffold lipids that enable the binding groove to be filled. If we superimpose all of the different structures that we currently have for the different isoforms, we can get a picture of the sort of shapes of the different lipids that can be bound in the grooves, and they vary quite differently. And actually, if you read the rather excellent paper that came out in Cell from Xiaoshang and Branch Moody and Graham Ogg and Jamie Rosjohn and colleagues, they've got a really good description about how these binding sites change between the different isoforms. And importantly, in here, there are differences in the sort of like the methylene unit count. So this is the size of the lipids, the number of carbons in the tails. And we see some distinctive isoform preferences in terms of size, and that's the sort of thing that we'll see in the next presentation. And we're also showing on the portal as well. If we look at how individual lipids sort of sit in there, we can see that they fit really rather well, and the spacer lipids pack the groove out. And actually, over on the right-hand side, we can see all the structures of one particular spacer lipid, and it occupies a pretty similar position in the groove. And this in some ways is quite homologous to some of the water molecules that you see in class 1 and class 2, where some of them are in very sort of standardized positions across lots of crystal structures, mainly the water molecules that hold the N and the C termini in place for class 1, and ones which help hold the backbone in place for class 2. So that's a quick sort of run through of CD1 isoforms and their structures. And I'll briefly talk about the lipidomic database. It's in development at the moment. There'll be another preview going up on Friday, assuming that I can get the hotel Wi-Fi working where I am. For some reason, it didn't want to let me join with my AWS account earlier, but we'll get that one fixed. And the database has two types of data in there. One type is the data from this paper, and that we'll be hearing about more in a moment. And the other is the crystallography data that I've been sort of showing you an overview. And so these are all structures of CD1 isoforms with different lipids that you can find on the protein data bank. We're pulling them together as a complete collection, and we're sort of indexing them in the same way as the lipidomic data. There are two distinct qualities of data, though, and it's worth understanding the differences between them. So for the lipidomics data, it's derived from mass spec measurements. What we have is a molecular weight, which gives us a molecular formula for the lipid. It may tell us how many unsaturations that lipid has, and gives us the overall methyl count for the lipid tails. However, we don't know where the unsaturations are, and we don't know the distribution of the chain lengths if there are more than one chain. And we don't get information about the spacer or the scaffold. This isn't remotely a criticism of the type of data that comes in. I think it's a miracle that we get what data we can from the mass spec on these lipids. And I know it was a very long piece of work to get there. And so it's just wonderful to have the more diverse data. There is data for well over 100 lipids from the lipidomics data, where we know all the details, which far outweighs the data that we have from the structures. From the structures, we know the exact lipid structure. We know where the unsaturations are. We know the length of the different chains that are there. And we know where there are exact spacers or scaffolds as well. And so we have two different data qualities. And what we're trying to do with the sort of portal is to sort of highlight that where we can, and to make it clear. So I'll stop sharing this particular window, and I will start sharing another one. Sorry, I'm just having a bit of a problem at the moment. Right, we're back. Sorry about that. Can you see this this browser screen? Hopefully you can. So this is the home page for the lipidomic database. The main data that you see when you come to the home page is the lipid class data. So this is the data from the lipidomics data set. If we pick a particular class, so a sphingomyelin lipid, we've provided some sort of simple example 2D structures. These are there largely for people who are coming maybe from a peptidomics background, and you can't imagine what a sphingomyelin lipid looks like. You know, sort of, it's quite useful me being the test case for this, because lots of my chemistry knowledge on lipids is several decades old, and it's useful to visualize what type of lipid it is. What we also see here is data that Xiaoshang will be talking about in a bit, which is the sort of like the isoform specificity data. So this is showing you which isoforms particular lipids preferentially bind to, and you can sort of click into one and find other lipids that are like it. What you can also do if you are somebody who maybe has some molecular weight data is you can search for specific molecular weights, and it will find you lipids that are closest to that. So if you've got an idea for a lipid that you have some molecular weight data for, but you don't know exactly what species it is or which CD1 isoform it might be bound to, or maybe you do have that specificity data, you can use the search to find that. And if all you really have is the number of carbons in your lipid, you can also find particular lipids that match that number of carbons. So there are different ways that you can do the searching. Jumping over to the structural side of things, and this is some work in progress, so excuse the fact it's a bit untidy at the moment. We give you some of the visualizations that you've seen earlier in this, showing you how the lipids in general fit into that particular isoform, how they protrude out of the hole and are accessible to T cells, and the general shape of the lipid populations for that particular molecule. What you can also do is jump through to individual lipids that are found in the crystal structure. It'll tell you the role of the lipid, whether it's an antigenic lipid or a scaffold or a spacer, and it will tell you which structure it's in. And then it'll also give you some machine readable representations like Smiles representations and Inchi representations, which you can then use either in docking software or other things like that. Now, obviously, we only have this sort of Smiles and Inchi format data for the crystallographic lipids because we know exactly where the unsaturations are in those. You can also look at if that particular structure has got other lipids in there in a spacer role. You can click on that and find out about other structures that contain that spacer. So this one, for example, is quite a common spacer, this dodecane lipid, and it's a spacer in CD1B, CD1C, and CD1D. And you can look at how similar the structure of that lipid is across those different structures. I should say that by the time of the hackathon, we'll also have machine readable data in the portal, so that there'll be a rudimentary API so that people can start playing with it. But what they can also do is they can start, as soon as the hackathon has produced some standards, then obviously we'll adopt those standards for all the data in the portal. So I'll just jump back to these other slides for a moment and just to give some acknowledgements. So I'd like to thank folks at the EBI, so my supervisor in Open Targets and the Protein Data Bank in Europe, Ellie and Samir, Hashem Kui and Graham Ogg's lab in Oxford, Branch Moody's lab in Harvard, and Jamie Ross-John, who's done loads of the structural work, who's at Monash. So thanks ever so much, and I'll stop now and happily take questions.
Thank you, Chris. I will monitor the Q&A as questions come in and present them to you. So if any of the audience has questions, please post them in the Q&A window. I thought it was interesting, Chris, that you, from my perspective, I thought it was interesting because your background where you studied HLA peptides or MHC-bound peptides and characterized those, and then you segued into characterization of CD1 and the lipid ligands. So if this is a presentation for the Society of Immune Polymorphism, we had group 1 is A, B, CD1, A, B, and C, and then group 2 was CD1D. So there's 4 different loci expressing CD1 molecules in humans. And I think you said there were 16, 28, and 21 alleles at each one of those loci for A, B, C, and D. Do you think those numbers, did I get that wrong?
I think you might have done, yeah. I probably wasn't very clear there. I think
No, I think I missed it.
no, I think what I was illustrating was the number of structures for those different this is always good user testing. So you've been, external user number 1, Willie, and it's brilliant to know where you found a flaw in our product. So what that was the number of structures for each of those different isoforms. So each isoform is monomorphic, and there are just a different number of structures for each one. And I think I will note that down as a thing to tidy up and make more obvious in the next version.
So what you're saying is at each of the loci A, B, C, and D, there's a single allele for the CD1 molecule. Do you think those numbers, if we just start there, do you think those numbers will expand?
I'm always willing to be surprised by anything to do with the MHC having more diversity than we currently think it does. That's always a joy. But I think similar to MR1, I think it is probably fairly monomorphic. There might be some, I'm not saying there wouldn't be. There could well be some alleles somewhere, but they've not been seen yet. And I think, sort of there are certain lipids that you want to make sure get presented by each isoform. So I think there are probably some selective pressures on it not having a polymorphic binding site, in the same way that MR1 is a very distinct molecule, has a very distinct set of ligands, and if you start messing with the binding site with polymorphisms, then you maybe would end up not being able to present the right sort of ligands.
All right, we've got a couple of questions rolling in from distinguished scientists. Dr. Curtis McMurtry at Pure MHC asks, given how the lipids fit in the groove, could you take known lipids from other databases and predict whether it would bind any given CD1 isoform, analogous to net MHC?
I think it is possible. I think the tricky thing is we're not entirely sure yet about the rules of the binding cleft, in the same way that we are with class 1. Sort of the lipidomics data has been very hard fought for and hard won. And up until we had that, we really only had sort of maybe, 10 to 15 to 20 known lipids for each individual isoform. And that's really not enough of a training data set that you need for something like NetMHC. So if you look at sort of NetMHC's training data set, it's horribly biased, as we all know. And you've got sort of 200,000 peptides for HLA-A0201, and then you've got sort of, decreasing number of peptides for sort of other alleles. But if you look at it, you've got around 1,000 peptides for quite a large number of alleles, for maybe sort of 60, 70 alleles. And that's what's needed to generate that sort of quality of prediction. And I think we just don't necessarily have the volume of data yet to make those sorts of predictions. I've been doing some testing with docking and seeing if we can recapitulate the structures using sort of docking algorithms, and we can pretty well. So we could, in theory, go sort of creating synthetic data sets at some point for it. But I think we'd really want a lot more positive and negative controls. I mean, if we remember the early days of NetMHC-Pan from Soren Boos's lab, they did a lot of binding assays, and they had both positive and negative data in there in the early versions of NetMHC-Pan. And I think we'd need that sort of data to go and create those sorts of predictions. It's something that Hashem Kui's group is interested in. They're experts in sort of machine learning, and they're looking into that. And there will be a hackathon next autumn in Oxford looking at sort of machine learning applications on different sorts of predictive data sets, antigen specificity and peptide binding and lipid binding and things like that. So I think it's early days, but it would be lovely to get to that place.
Well, I know that Curtis and I know that you will be at the hackathon next month in Oklahoma City. So perhaps we can get a start that'll segue into next year's hackathon to further digress on that. I have another question here from Dr. Jim Kaufman, another distinguished MHC scientist. She said, thanks, Chris, for the great summary about CD1 isotypes, which bind particular kinds of lipids. Isn't it true that the lipids in the two chicken CD1 molecules are quite different from humans and mice? And if so, do you have any sense for why certain kinds of lipids are useful?
Thanks, Jim. And I owe you an email, so sorry about that. But as ever, a really tricky question from Jim. I don't think we know yet, Jim. I'd love to sit down and chat with you about this. I sort of do wonder whether, because lots of the targets for CD1 molecules are things like NK T cells and gamma delta T cells, and the gamma delta T cells sort of sit to the side when you look at the structures. They don't sit on the part where the lipid head groups is, whether some lipids are there for sort of almost homeostatic reasons, or for sort of sensing whether there's stress on cells or things like that. And others maybe are more for sensing lipids that come from bacteria, or things like that. So it would be really interesting to look at the differences between the human and the mouse lipids, and look again at the different cleft residues, because there are some differences between chicken and mouse CD1 compared to human CD1. So I think it would be interesting to, and again, this might help us with these sort of eventual NetMHC pan-like predictions, is to expand the data out and to also make it more varied. I think, with all machine learning things, you struggle if you've got very sort of homogeneous data. And I think, sort of expanding it out to chicken and mouse would be potentially very useful. So I'd love to chat with you about that, Jim.
And maybe if you could recruit Jim to attend the hackathon in Oklahoma City, because I think it was just actually when COVID was just kicking in, he visited in February of 2020, he visited, and that may have been our last visitor for a year or so. And Jim says, thanks so much, Chris. I don't see any more questions in the chat and the Q&As. And I think our time is pretty good. We're almost to the halfway through the hour. So Chris, thank you very much for your presentation. Thanks for taking the time to do it while you travel. And I'm glad it technologically worked out well. All right, our second speaker today is Shouzhong Huang, and the title of his talk, I'm going to give him a brief intro, is the Elutional Human Lipidome. Let me go and pull up his bio sketch real quick here and give you a little bit of introduction. I was really impressed when you look through his bio sketch. He has a bachelor's from Beijing University, a PhD from the Ohio State University, and he did a postdoc at Washington University and then a postdoc at Brigham and Women's Hospital, Harvard Medical School. And when you look through his publications and his work, you really see that Shouzhong has done a tremendous job of collaborating with those in the field. And for instance, I just went through his bio sketch and I see, and these are just people I recognize. I sincerely apologize to those that are heavy hitters in the MHC field that I didn't recognize, but I see Jamie Ross-John, Branch Moody, David Lewinson, John Altman, David Fremont, Ted Hansen. You just see a list of Steve Percelli. You see a list of heavy hitters scientifically that Shouzhong has collaborated with in order to build a very impressive resume. And following all of those collaborations and the two postdocs, he is now an associate professor. He has his own group at the Texas Biomedical Research Institute in San Antonio, Texas. So without further ado, perhaps Shouzhong, you could proceed with your presentation. again, and my computer is messing with me here, which is titled The Elutional Human Lipidome. Perfect.
Okay. Yeah, thank you, Willie, and thank organizer for invitation. So I personally working in Texas Biomed Research Institute now, but this work has been done with the Branch Moody in Brigham Women's Hospital. So first, I would like to acknowledge the collaborators, including members in Branch Moody's lab, and also members in Jamie Ross-John's lab in Monash, and from Grant Ox lab in Oxford, and Adrian Manas's lab in Netherlands, and also thanks to NIH Tetramer facility and John Adman for CD1 Tetramer. So we know that elutional antigen peptidome or lipidome are so critical for T-cell antigen discovery, right? So back to early 2000s, we can see the peptide profiling for MHC2 molecule from Unanime's lab, and epitope discoveries for West Nile virus from Curtis, actually, in Willie's lab, and a single peptide MHC complex compared to wild-type KB molecule from Ted Hansen Connolly's lab. And for the CD1 lipidome discovery, we can go back to the 2009 with a few papers, one from Creswell's lab looking at the different subcellular compartment lipid association with the CD1D protein and a very comprehensive cellular lipid profiling for human CD1D protein from Willie's lab as well. And myself working with Branch Moody's look into deoxyceramide and diacylglycerol as a CD1B scaffold lipid at early time. So CD1 expressed in many different places. I want to bring out this slide, just very quick overview for different CD1 expression. So we know that CD1A as a marker for Langerhans cells. So we can also see CD1A expressions high in the skin psoriasis. And CD1B is a strong marker for dendritic cells. So we can also see CD1B expressions in alveolar macrophage in COPD. So can you see my mouse? Or I can use.
Let's see. I'm not seeing it right now.
Okay. Let me use a laser pointer.
There it goes. Looks
okay.
good.
Okay. So we can, for CD1C, it's a really strong marker for dendritic cells. We can also see that CD1C expression in the marginal zone B cells. And CD1D, we know that they express them in the mucosal epithelia and thymic cortex, but in the liver cancer, we can see their expressions in hepatocyte. So the question is, we know that there are some tetramers standing for invariant NKT cells, but actually we're missing markers for many other CD1 restrict T cells. Therefore, lipid ligands certainly is important for tetramer detections to develop in the future. So lipid reactive T cells mostly restricted by CD1 proteins. So we know that invariant NKT cells versus germline encoded microglial lipid reactive T cell, they express the invariant T cell receptor. But many other CD1 restrict T cell, they express variable T cell receptor, and CD1 proteins. We have different lipid binding cleft. For example, CD1A and CD1D, they have the A prime pocket, F prime pocket, but CD1B and CD1C, they are very different. CD1B, you have a C prime, additional C prime pocket with a T prime terminals. And CD1C, you have open structure of the F prime pocket with a more flexible binding with a lipid. And with a heterogeneous CD1 restrict T cells and different structures of CD1 proteins, we're going to ask the questions whether these CD1 protein can bind to different or similar lipid species. If they bind to a similar lipid species, whether they have quantitative difference, and whether some CD1 isoform can enrich certain endogenous lipid, and whether size-based motif can be discovered for lipid binding to CD1 isoforms. So to answer these questions, we try to to bring out a CD1 lipidomic platform. So we first express CD1 protein in human cell line, then purify the protein, extract lipid, then do the mass spec profiling, then analyze those lipid profile by looking at their share or unique molecule features by different CD1 isoform. Then we identify this lipid and generate hypothesis regarding the size of this CD1 lipid for different CD1 isoform. Then we test this hypothesis. And to express the CD1 proteins, we generate two different formats of the CD1 recombinant CD1 proteins. First format we call linked CD1 protein with linkers in between a beta-2M and also heavy chain. The second format is generated by NIH tetramer facility, and they use a zipper to link the heavy chain and beta-2M together. So we also use the MSC class 1 as a control, and HLA-B27 and MAMU A01. So we express these proteins in two different cell types, including myelogenous human cell lines, K562, and also embryonic leukemic cells, 293 T cells. So then we purify the protein and extract the ligands using photo extraction methods that's in profile these extract ligands. And certainly this is, we know that this is a engineer linker linked protein or zipper protein, but we also confirm a similar result with a tail intact CD1 protein in monocytic cells like THP1 cells. So elution of CD1 lipid, we need to answer the question whether those lipid elute from CD1 protein, that they are still stimulatory. So then we took the two cell lines that restricted by CD1A protein. BC2 cell lines restricted by CD1A protein for presentation of the self-ligand, and CD8 cell lines stimulated by DDM, which is the mycobacteria, dideoxymycobacterium antigens. So then we feed this eluted CD1 ligand to the cell line expressed CD1A protein co-culture with this T cell line by mixing up self-ligands versus DDM molecules. We can see if we just provide a self-ligands elute from CD1A protein, we can see they activate this is BC2 T cells. So with enhanced concentration of the mycobacterial ligand, we can see this mycobacterial ligand can compete away these self-ligands that then inhibit the BC2 activation, but enhance the CD8 cell response. So therefore, eluted CD1 ligands can activate T cell and can compete with mycobacterial lipid antigen. So that provides a really nice and functional pool for us to further profile the lipid molecule from this pool. And certainly, so we use two different cell types and two different formats. Then we do a mass profiling and looking at a molecule's features. We define a molecule feature as these features can have three different parameters. One is MZ values, intensity, and also retention time. They all together define each single molecule features, for example, showing here as one single P through our different retention time. So we can analyze these molecule features using volcano plot to show that a lot of these molecule features are strongly associated with a linked or unlinked CD1 protein, but not with MSC, HLA class 1 molecules. So this is a vertical green line show the P values, and vertical green line show the full intensity compare between different association with the CD1 versus HLA class 1 protein. Then we can further compare whether these molecule features are more shared by different CD1 isoform or is unique for each single CD1 isoform. Surprisingly, we found that the majority of the more than 50% is 60% of molecule features are shared by all 4 CD1 protein, and many of them also shared by 2 or 3 CD1 isoforms. So therefore, we want to focus on these shared molecules to look at whether their structures identities. Simply go through this identification approach. So we identified each single lipids based on the collision patterns that's matching with a predicted fragment as we show here. For example, in here we have a signature predicted fragment containing the ether link fatty acyl chain that show as, for example, here's a 563 over here, and so this highest peak represent the choline and the phosphate group. And there's a very nice parallel match with a synthetic reference compound we purchased from the commercial company, and the patterns are very similar. So based on these methods, we can identify a series of the ether link phosphatidylcholine throughout similar retention time. Suddenly we can see the longer of this anchor size have a little bit longer the retention times represented by polarity of the HPLC gradients. So using these methods, we identify more than 300 lipids from 14 families as a CD1 ligands. The question is from this pool, whether we can identify the signature lipid motif, for example, the size motif, and represent over here throughout the different retention time, and polarity is represented as a polarity. We show this color-coded lipid is being annotated, but there are many others which still remain to identify them. So these are including a different ceramide and sphingolipid, and also phospholipid, and also glycolipid as well. So we want to focus on very abundant species of phosphatidylcholine and ethanolamine and sphingomyelin to look at the size differences. We define the size based on head groups, so based on the lipid anchors show as red colors. You can see these red color lipid anchor can be calculated by the methylene unit or carbon numbers. If we show that, for example, this binding of a phosphatidylcholine with different CD1 protein, right? From the smaller size, from the smaller mass to higher mass, with the small anchor size to the higher anchor size. We can see these are color coding in the middle range. You can see a lot of association of this lipid with a CD1B protein, but in the higher anchor size, more association with the CD1D. So that's a really interesting phenomenon. We can expand it to the analysis of the different families of lipids. If we calculate the Z-score, which is represent the normalization of the intensity of different lipid in the same lipid species, right? Normalized with the mean and standard deviation, we can find that the CD1 A, B, C, D, they have different pattern of the association with lipids. For example, over here is the PC, and the higher anchor size, we can see that more associated with CD1D, but CD1A is in the middle range, and CD1B and C at the lower range. So according to calculations of many different structures from CD1 A, B, C, D, and their predicted mean anchor size can be like 38, 65, 47. But actually, we detect from our profile lipid size, we can see CD1A certainly is matching the predicted anchor size, but CD1B is way off. We call mismatch with the anchor size. So then we hypothesize maybe we need a too small lipid in one CD1B protein. To test this hypothesis, you can see that the endogenous lipid bindings in the crystal structures, you do see two lipid species in one ligand binding group. So you can see scaffold lipid sitting below, and antigenic or amphipathic lipid sitting on the top. And certainly if you load it with a very long lipid with 80 methylene units, glucose monomycolate, and you can only see one lipid associated with a lipid binding cleft. So can these dual lipid association explain lipid exchange throughout the CD1 trafficking intracellularly. We know that CD1 trafficking through a different compartment of the endocytic compartment. So one question is whether CD1B, when they exchange it with the exogenous lipid or some other endogenous lipid, and how that impact the dual lipid binding. So to answer these questions, we can see endogenous lipid, if we feed additional exogenous lysosulfatide or exogenous sphingomyelin, exogenous phosphatidylcholine, we can see the replacement only happen on the top lipid, the upper chambers, for that amphipathic lipid species. But the scaffold lipid still remain, right? So if we replace it with a longer lipid, a C80 glucose monomycolate, they're going to replace both upper and lower lipid. So that possibly can represent one scenario in the endocytic pathway how lipid exchange occurred. So to further understand whether this dual lipid binding will contribute to T cell activation. So we take advantage of the T cell line that respond to glucose monomycolate, C32 glucose monomycolate. And we can see whether phosphatidylcholine, if they sitting on the top, can compete with this glucose monomycolate for T cell activation. And this is also based on our early discoveries on diacylglycerol is a non-polar, less polar lipid that could be sitting on the bottom of the CD1B ligand binding group. So then we design an experiment using plate-bound CD1B protein and to put together with a glucose monomycolate reactive T cells and throw in DAGs, diacylglycerol and PC and C32 glucose monomycolate to allow them to compete each other. So results show very interestingly that if we only put in diacylglycerol, they don't activate these T cells. So if we increase the concentration of the phosphatidylcholine, they're going to compete away this glucose monomycolate that inhibit a T cell response. But if we added a different concentration diacylglycerol, we can see at a certain ratio, a certain concentration, you're going to see the enhancement of the glucose monomycolate stimulate T cell response. So these experiments interestingly support that this scaffold lipid could have a functional impact on T cell response. So I'm going to quickly summarize that. So in this talk, I already talked about the elutional cellular lipid can regulate mycobacterial lipid for T cell activation, and CD1 isoform can sample heterogeneous cellular lipid and based on size. And CD1B can capture dual cellular lipid, and CD1B can exchange a single or dual lipid, mimicking antigen loading intracellularly. And CD1B enriched scaffold lipid can enhance the T cell activation by mycobacterial lipid antigen. So I'm going to stop here and take questions. Yeah, that's it.
That was a great presentation, and really enjoyed viewing the complexity of the CD1 lipid ligands and how they compare from one molecule to the other. I have several questions, so I might start with those, and we'll see if any accumulate in the Q&A. And I don't know if I'm asking a question that shows my bias or my age, but as we were talking with Christopher earlier, I'm looking at the lessons we learned in peptidomics and seeing if they might not apply to the lipidomics. Do you think, or do you feel that analyzing the ligands presented by CD1 of primary tissues is essential? I mean, do you think that you're going to, or the field's going to be led or pressured or go in the direction of getting a tumor tissue or getting primary cells? And do you think we'll need to or be able to look at lipid ligands presented by CD1 of primary tissues?
Yeah, I agree with you. That's a really important question, actually. So when people look at the liver tissue, right, they look at the environment versus a variable NKT cell. They like to know the complexity of lipid molecule in the liver tissue, how that contribute NKT cell response. And I think regarding methodology is somewhat difficult. For example, we don't get a lot of CD1 protein, right, from liver tissue. And maybe we can do in a way like we can chunk up the liver tissue, see which one stimulate the T cell response. And then we can go after that result, right? So I think it's a very important question. Sure, in the future, with a high resolution, high sensitive mass spec instrument coming out, sure, that's should be able to become reality.
And follow up on that, when you look at unsaturation, when you look at the double bonds in the acyl chains,
Mhm.
I haven't looked at lipid ligands mass spec data for a few years, so I'm a little bit behind speed on that. Is it difficult? Is there any challenges to assigning the double bonds or the unsaturation, the positions on the acyl chains and the cis trans assignment, or is that's now fairly robust?
Yeah, I think certainly first that you are pioneer doing like a CD1 lipidome as well, and for the assigning double bond, recently came out a new technology called UVPD, ultraviolet photo dissociation. I think that would be really helpful to looking at a position of the double bond. Yeah, maybe many others approaches are also available, but this is possibly more forward approach. Yeah.
Okay. We've got a question from Dr. Jim Kaufman. Thanks so much for the lovely talk. Given that different isoforms of CD1 bind different kinds of lipids, do you have a sense for how well the polar head group recognized by the T cells correlates with the kind of lipid? That is to say, does the size and character of the lipid part determine what kind of polar head group will be recognized by the T cells?
Yeah, that's at the early findings regarding CD1D to NKT cells, the general hypothesis, a lot of evidence also support that head groups interact with the T cell receptor, right? Because they're sitting on the outside. And for the lipid, for the fatty acyl portions, usually go inside for the CD1D lipid binding. And head groups, possibly importance of head groups also varies among different CD1 isoform. For example, CD1A, right? And you don't need a hair group to be recognized by T cells. For example, about 10 years ago, Branch Lab published a paper on the wax esters and also the skin lipid, the oily lipid is nonpolar, then can sitting inside a CD1A protein that still stimulate T cells. Yeah, that represents another angle of the T cell lipid interaction. Yeah.
Okay. All right, I don't see any other questions. So we thank you again for taking the time to present to us. You know what, I'd follow up with one quick one since we have a couple minutes. Again, for the Society for Immune Polymorphism, I was wondering when you began your studies of the CD1 lipid ligands, and now that you've progressed, are you surprised at the number of different lipid ligands? I come at everything from an MHC perspective, but if you come from a lipid perspective, if you come from a lipidomics perspective, how many different lipid ligands do you feel will need to be sampled? How much complexity and heterogeneity in the acyl chains? How much heterogeneity in the head groups that those acyl chains are linked to? What do you think is important for the CD1 molecules to present in return in terms of variety?
Yeah, yeah, right. That's a really surprising. I think in the very early discoveries by a science paper, and they identified one single molecule, right? One single lipid. I think it's a phosphatidylinositol for CD1B protein. And actually, when I read your paper, when I read your paper, it's published in 2009, and I was surprised about wow, so many lipid binding CD1D protein. And then I listened to the talk given by Jamie Rossjohn in one NKT cell mini, and he showed that I think it hasn't been shown before that time. He showed that a lot of endogenous lipid can be co-crystallized with CD1D protein, and then suddenly opened up the mind that oh, this CD1 protein may bind to many different lipids, but we still don't know their functional implication for each different lipid. That leads back to the James question. So we still need to do a lot to understand the functional implication for those different heterogeneous binding. Yeah.
All right. Thank you very much for taking the time to present today.
Thank you.
We look forward to seeing you in Oklahoma City for the hackathon.
Yeah, thank you. Look forward to it.
And I'll turn this back over to Martin Myers right now. Martin, do you have any additional information to pass on?
Just wanted to say, in case people joined through later in the webinar, that the hackathon is in September, and I'm hoping people can make it. This is Tuesday, the 17th of September through Thursday, the 19th. And if you send me an email or go to the website, I've put it on the SIP website too. Yeah, I'm hoping we'll and we'll put this video online so other people can see it later too. But yeah, we're hoping to figure out. I mean, I can see some data standards opportunities here already, and more questions than answers, it looks like at this point, about what we're actually going to do at this hackathon. But looks like we have some data and we have some people that know stuff, so that's a good start. So that's it. Thanks everyone, and thanks for staying on time, and I really appreciate the talks. They were I learned a lot. Thanks everybody.